Thank you, Dr. Resnick, for that very kind and elaborate introduction, a bit over the top. As I often say, I put on my pants one leg at a time like everyone else, so I don't feel as important as Dr. Resnick ascribes to me. I'm going to try to wrestle today with the conceptual problems that are brought up by two fundamental issues that the CBTN must confront, to my mind, and I will try to deal with these at the conceptual level. And the fact is that the CBTN focuses on childhood cancers and on brain tumor cancers. And as I will try to indicate, there's some fundamental logical disconnects between childhood and adult tumors, as well as there being differences between the tumors affecting the brain and those affecting all the other organs throughout the body that have been more intensively studied. Given that this is a very heterogeneous audience, I will try to make this as understandable as possible. My teaching with Eric Lander, as was mentioned before by Dr. Resnick, certainly introduced me to the rigors of teaching freshman biology, and I hope that helps me. It turns out that Dr. Lander ended up better than I did in life, since he is now President Biden's science advisor, whereas here I am in the wilds of New Hampshire talking to you about cancer, but I have no regrets. That having been said, let's just discuss some of the major conceptual issues at hand as we move toward the hallmarks conceptualization. Here is one fundamental disconnect that we need to deal with. Here is the onset of adult cancers in both men and women. You see that. And with the exception of breast cancer, the other kinds of cancers are diseases that require 4, 5, 6 decades of life to develop. And we know, at least in one case, how long these steps take. Here's the global cigarette consumption together with increases in lung cancer deaths. And here we see it takes about 30 years for an adult tumor to develop. Again, I will return to this theme later, but this begins to highlight the fundamental logical or conceptual disconnect between adult tumors and what one deals with here. Much of our thinking about adult tumors has been conditioned by the work of Bert Vogelstein and colleagues at Johns Hopkins, in which already in 1989, almost a third of a century ago, he connected the histopathological changes in colorectal cancer progression. You see those changes below, and you can see the micrographic histopathology above. He connected those successive changes with the acquisition of distinct genetic alterations in the cells that were participating in these various stages of tumor progression. Thus, for example, early on, normal epithelial cells acquired a loss of heterozygosity in chromosome 5. In the adenomas in the middle, they acquired another change, an activation of the RAS oncogene. Later on, they acquired a loss of heterozygosity in chromosome 18. And in all of these cases, this represented genetic changes in the evolving cells that one imagined were actually driving forward the multi-step progression of tumor. In this case, to be sure, in the context of the colon and colorectal carcinoma. Indeed, here to be more specific, the first conversion required the loss of a tumor suppressor gene called APC. Here we have the activation, another genetic alteration of the KRAS oncogene. And here's the loss of yet another gene, P53. And each of these changes involves a genetic mutation. That is to say, these mutations, which are acquired successively, are important for driving this multi-step process. They are more than just incidental markers of this process. These are probably the engines that drive these steps forward. And each of these steps, as indicated here, 4, 5, 6 steps, requires many years, often between 5 and 10 years to reach completion, so that most of us, thank the good Lord, reach the age of 60 or 70 or 80 without these multiple steps having reached completion. Completion in this stage would be a carcinoma, as you see here in the light brown, which would be a primary tumor, and invasion and metastasis. These are, to be sure, environmentally driven. And as I often say to students, if you'd like to have this accelerated, you could go to Wendy's or McDonald's and eat there every day. And the 5 to 10 year time span between individual steps here could probably be compressed to maybe 5 years. And the process as a whole could be accelerated. Here we have another discrepancy with the childhood brain tumors that you're focused on, because to my knowledge, there is virtually no information indicating that there are any environmental signals, in this case, in the case of colorectal cancer, a diet being important determinants of the appearance or the lack of appearance of a given tumor. Here, I just extend what I showed you before to the context of pancreatic carcinoma, wishing to generalize it by saying that in the context of the pancreas, there are also multiple distinct histopathological changes shown above, which also involve, as you can see below in these various labels, also a whole series of distinct genetic alterations. So there is a generalizable paradigm, most developed, to be sure, in the context of colorectal carcinoma pathogenesis and here in pancreatic, but presumably occurring also in many other kinds of adult tumors, again, tumors that require many, many decades to reach completion, reach fruition. And implicit in the thinking that's going on here is the notion, the conceptual model in everyone's mind, that one starts out at the top with a population of normal cells, one of which in the red sector you see here acquires an initiating mutation, a mutant gene, an oncogene. And this initially acquired mutation enables that mutated cell to spawn an entire cohort of descendant cells, maybe they being able to grow more rapidly than the normal cells, and ultimately a whole cohort of descendant cells, as many as 1 million cells, in one of which, stochastically, a second mutation may take place. And now we have a doubly mutated cell that grows even more rapidly than its singly mutated counterparts. And so we have a second clonal expansion, where now the doubly mutated cell spawns a descendant cohort of a million cells once again, in which a third mutation and so forth can occur. And this type of Darwinian evolution is imagined to occur in the context, in this case, of the human colon, or in virtually all other adult epithelial tissues, to explain how multiply mutated cells actually arise and how they can acquire these otherwise rare mutations. Because I mentioned that many of these mutations occur with a probability of only 1 in 1 million per cell generation. Finally, as these multiple mutations are acquired, the tumor progression accelerates, and toward the end, one begins to have multiply mutated subpopulations indicated here in the lower right that accumulate more rapidly than Darwinian selection can eliminate less fit versions. And so there's a great diversification intratumorally observed in the subpopulations of cells within a given tumor that differ subtly from one another in specific mutations they happen to have acquired during the course of multi-step tumor progression. I now shift gears momentarily to ask an experimental question. Given what I've just told you, how many genetic mutations are actually required in order to convert a normal cell into a tumor cell? This bears on what I've just been discussing, as you'll see momentarily. And here we see experiments done in 1983, where one took rat embryo fibroblasts, introduced into them via gene transfer, either a MYC or E1A oncogene. Nothing happened. Alternatively, one introduced a RAS oncogene via gene transfer into cells over in the right, and nothing happened. But if the two were concomitantly introduced together into rat embryo fibroblasts, one could observe the outgrowth of foci of transformed cells, which established the paradigm that oncogenes, or genetic alterations, can collaborate with one another to change the phenotype of specific cells. And here one saw one example of that. To be sure, this paradigm was violated when one began to look at human cells, because between 1983 and 1999, no one had ever taken a normal human cell, and transformed it experimentally into a tumor cell. Human cells were widely believed to be impregnable, ironclad, and untransformable. And the reason this was so was that there was one missing ingredient in the cocktail of mutated alleles that were introduced into human cells, the missing ingredient being the telomere, or the H-TERT telomere repairing enzyme. And here in work of Bill Hahn in 1999, one saw that actually 5 distinct genetic alterations needed to be introduced into a normal human cell in order to convert it into a tumor cell. One may ask, by the way, how this contrasts with what I showed you just before. And the profound difference is that rodent cells require only 2 cells, and human cells, rodent cells require only 2 genetic alterations, whereas human cells require 5 genetic alterations. And this is an evolving observation. It turns out that among mammals, as we mammals evolve to greater and greater complexity, longer lifespans, greater body mass, and many more cumulative cell divisions in a lifetime, we needed to develop cells that were more and more resistant to transformation. And as a consequence, here one saw that these required mutations, in this case, the number being 5, was a reflection of the fact that the regulatory circuitry in individual human cells demanded at least 5 distinct changes before the cells would become fully neoplastic. This, by the way, begins to echo and reveal the profound disconnect with pediatric cancer and the fundamental unsolved problem of one of them of cancer biology, to my mind, one of the most provocative, and that is why and how pediatric cancers can develop in such a short period of time. The paradigms I'm showing you here reflect adult tumors, and yet there's a fundamental different biology that is poorly understood at any level, which says very clearly that early embryonic and maybe even pediatric cells within a human require far fewer changes in order to undergo neoplastic transformation. And therefore, it is my belief, my firmly held belief, I echoing what I say almost once a day, people who are convinced of things are often wrong in a loud voice, but I am convinced that there's a fundamental problem here which remains unsolved, which is the following. As the human fetus develops, one begins to undergo a profound shift in the biology of cells where they are initially relatively transformable, let's say with 2 alterations, more like the rodents, and require ultimately more genetic mutations. This is not often discussed, but to my mind is one of the fundamental problems of modern experimental biology. How can we conceptualize the possibility that somehow there is some biological shift which occurs throughout the body of a developing human or fetus that changes its cells from one state, which is less resistant to transformation, to the adult state where it's more resistant? And I just mentioned to you the fact that we as adults go through 10 to the 16th cell divisions in a lifetime. A fetus goes through about maybe 10 to the 11th, comparable to the number of cell divisions that a mouse experiences in its lifetime. And it's therefore my belief that the relative transformability of a fetal and early life cells reflects the fact that these cells and the organism that carries them, that is the developing fetus, goes through far fewer cell divisions, realizing the fact in both cases that cell divisions represent occasions for inadvertent genetic mutations. The risk that we adult humans confront being indicated by the 10 to the 16th successive cell mutations that each of us goes through in an adult lifetime. Now, clearly, what I'm indicating here is some kind of correspondence, because this oncogene collaboration assay, in this case requiring 5 genes, is reflected in the requirement I showed you before. That is to say, one presumes that these 5 signaling pathways must in one way or another be perturbed through genetic alterations in order for a normal human cell to become a tumor cell. Again, there's not rigorous evidence that these 5 changes are required in all types of adult cells, but I think that it's roughly correct. And again, no one has any idea how to relate this to the relative transformability of pediatric tumors. Still, to be sure, there are similarities between adult tumors and pediatric tumors, or at least central nervous system tumors. And I indicate some here. Not all of these are ones that you deal with, but I'm just indicating these here to indicate the familiarity of some of these mutations to a researcher like myself who works exclusively with adult tumors, because to state the obvious, many of the similar mutations are operative in both. And so there is some commonality in how one thinks of the pediatric transformation at the biochemical level, maybe not at the biological level, but at the biochemical level in terms of the involved genes. Here, once again, is a reflection of this fundamental disconnect that, again, I admit I have no insight into. This is just the age of onset of a whole series of different kinds of human tumors. The brain cancer I'm showing you here is obviously not the type that is seen in pediatric patients, and it has an onset, a peak of occurrence at the age of about 60. Most carcinomas in adults, as I showed you before, appear first at the age of 60, 70, or 80. And here's the outlier, retinoblastoma. This is rather awkward for people like myself who try to explain how it possibly can happen. How is it possible that in the eyes of children, there can be 5 different mutant alleles acquired? The answer to this is there are not. There may be 1, there may be 2. Indeed, it is even conceivable that the knockout of the RB gene, which is obviously lies at the heart of the pathogenesis of retinoblastoma, may on its own suffice to create neoplastic cells. And again, I confess my inability at dealing with this conceptually. How can you get a cell transformed with a very small number of mutant alleles? And by the way, the reason why retinoblastoma shuts down by the age of 77 or 8 derives from the fact that by that time, the retina becomes a totally post-mitotic tissue in which, therefore, retinoblastoma pathogenesis can no longer occur. Here are some of the familiar markers. I took this from the Journal of Neurosurgery. These are all the familiar actors that operate in central nervous system tumors. You see them all here. Virtually all of them have been learned or gleaned from research in adult tumors, but clearly there's some involvement as well, equally in pediatric tumors. And we can see that as well here in medulloblastomas, with which many of you are familiar, where many of the losses that are observed actually are reminiscent or echoing what one sees in adult tumors as well. But still, as I argue time and again, the number of these changes that operate seems to be much lower, much smaller than what one sees in adult tumors. To be sure, I don't know, and maybe some of you do, whether the number of mutations in certain kinds of pediatric brain tumors are more than others. Do medulloblastomas require more genetic changes in order to become neoplastic than do gliomas? Again, that may be known. It's not known to me, but to my mind, that's conceptually a very important issue to deal with. Here, by the way, one notes metastases, which occur relatively infrequently in brain tumors, ostensibly because, as I've always been told, most brain tumors kill the patient long before metastases have a chance to insert themselves elsewhere in the body. So here are the much heralded hallmarks of cancer. You may have read about this in your preparatory material. To give you some background to this, in 1989, Doug Hanahan and myself were hiking, or no, 1999, we were hiking down the mouth of a volcano in Hawaii. He and I, both being alumni of MIT undergraduate education, said to ourselves, you know, we work in a field which seems to be a massive inchoate phenomenology. In fact, in my own case, in the early 1980s, a experienced veteran cancer researcher said to me sternly and in an admonishing fashion, said to me, he said, I should never confuse cancer research with science. Oh, and that stuck in my craw because to my mind, one needed to have some kind of conceptual structure with which to conceptualize the entire logic of cancer pathogenesis. So Hanahan and I wrote this review article. And we wrote it not to impose our will on the field of cancer research, but just to clarify our own thinking, and fully believed, like all other review articles that we'd ever written since and then, that this review article would disappear like a stone thrown into a quiet pond. But that's not what happened. If you look below, I just checked that this morning, there were almost 36,000 citations of this. Does that mean that this article is so powerful in its conceptual structure? Or, as I often ask myself, does it mean simply that by citing this article, the authors of many different the authors of many different publications simply obviate the need to look up lots of different references and therefore cite Hanahan and Weinberg as some facile way of sparing themselves work? Nonetheless, there is some utility in thinking about there being distinct biological traits that are associable with cancer cells. And when I say biological phenotypes, as I highlight above, I want to make a very clear distinction. These hallmarks are not indicative of any information about the genetic alterations in cancer cells. They are discrete cell biological phenotypes that are associable with the process of tumorigenesis, independent of how these biological phenotypes are achieved. And that's very important in thinking and conceptualizing the import of these various changes. Here was the 2011 version of this. Doug Hanahan called this the next generation. I guess that came from Star Wars. And we added some here, emerging hallmarks. And these you can see were deregulating cellular energetics and avoiding immune destruction. And these are still matters of some interest and debate, unclear in my own mind. Avoiding immune destruction seems increasingly to be important to the pathogenesis of many adult human tumors. That is to say, they undertake a whole series of strategies in immunoevasion in order to avoid elimination by arms of the adaptive and innate immune system. So this would seem to be a widely applicable trait. And deregulating cellular energetics. We all know since Warburg that the energetics, the metabolism of cancer cells is quite different from most normal cells. Indeed, the energetics, the metabolism of cancer cells is actually closely approaches rapidly proliferating normal human cells. So it's not as if their metabolism is in fact so different from one manifestation of normal cell metabolism. Still, unclear in my own mind, even though it's inserted here in the top left, is the question: is it essential for the metabolism of a cell to be altered, deregulated, in order for that cell to become neoplastic? And I still don't have a clear resolution of that in my own mind. Is altered metabolism an accompaniment of neoplastic transformation or a critical causal factor? Below, you see enabling characteristics. And here the distinction is made is these are not necessarily phenotypes of cancer cells or of tumors, as described before. Instead, these enabling characteristics are processes that make it possible for cancer cells to acquire the distinctive phenotypes we associate with them. Genomic instability and mutation. This seems to be critical simply because in a normal adult genome, the rate of mutation acquisition is so low that it's mathematically inconceivable that a tumor could form in a human lifespan. And so some degree of genomic destabilization in the terms of defects in cell cycle checkpoint control or in repair seem to be important in expediting the formation of an adult tumor. To the lower right, we see tumor promoting inflammation. It turns out to be the case that in the instance of for example, carcinomas, there's increasing evidence that almost all of them arise in microenvironmental areas in adult tissue where there has been chronic inflammation. And so this chronic inflammation may be analogous to tumor promotion as studied over the last 50 years. And it seems to be an essential ingredient to enabling many human carcinomas to arise, but is not itself an intrinsic part of the product of this, which is to say the fully formed neoplastic cell. So these are enabling characteristics rather than, in the strict sense, hallmark characteristics. People ask, well, do we want to add other items, other hallmarks to this list? And my response is, I don't know. And the last thing that Doug Hanahan and I ever wanted to be was to become popes who sat on some kind of throne and dictated what should or should not be the thinking of others. This was thrown out for public consumption, and we are not about to give our imprimatur on the correctness or the lack of correctness. It's simply to provoke people to suggest and to construct their own models of how cancer arises. As I've often thought and even said, the worst thing that can happen for any field of research is for a small number of so-called thought leaders to impose their will and their thinking on everyone else. And that's the last thing that either Doug Hanahan or myself intend in this. This one cited even more, evoked even more citations, 51,000. Again, I respond to that cynically by thinking it saved a lot of people work in having to look up various kinds of references. Now, to be sure, there is some interrelatedness between the acquired capabilities you see here on the left. These are really hallmark attributes and distinctive genetic alterations, the different phenotypes, the different genotypes I told you before. Here are, let's say, 5 different genetic changes, which I said might be involved experimentally in converting a normal human cell into a tumor cell. The intellectual challenge is to interrelate the list on the right with the list on the left. How do cancer cells actually acquire these hallmark attributes? And here's one attempt at trying to interrelate them, realizing that almost all of these genetic alterations have perturbing effects on multiple distinct cell biological phenotypes. You can see the list on the right and the list on the left. Still, there is some kind of interconnection between the distinct alterations at the genetic level that cancer cells require and the biological phenotypes that they must acquire in aggregate in order to proliferate as fully formed cancer cells. There is one interesting thing that's of special interest, I think, to pediatric cancer, maybe not to CNS cancer, and that's the following. Here we focus on telomerase, which is, as you may know, as you do know, an enzyme which is involved in preserving the telomeres at the ends of chromosomes and preventing their collapse, which must occur inevitably as cells go through multiple successive cycles of cell division, cell growth and division, and ultimately these telomeres collapse, which precludes the further proliferation of cells. Is this at all important for cancer cells? Well, in fact, in order for adult human cancers to thrive, they must develop a mechanism to prevent the further erosion of telomeres. Here we indicate this under the rubric of immortalization, and that that a cancer cell must confront the need of turning on telomerase, let's say, in order to begin to restore the length of its telomeres at the ends of its chromosomes. The beginnings of that is indicated here. Is this at all important for clinical cancer? So here are 2 kinds of pediatric cancers which are of interest to you, maybe, neuroblastoma and Ewing sarcoma. And if you look here, what one sees is a profound difference in the long-term clinical progression of patients, neuroblastoma patients. I know it's not in the central nervous system, so I have some degree of sophistication here, but between those whose tumors are negative for telomerase and those that are positive. And the same can be said of Ewing sarcoma. So at least one of these hallmark traits which is involved in telomere maintenance and the resulting cell biological immortalization, seems to be clinically important in determining the long-term outcome of tumor progression. In the end, one would like to be able to interconnect these cell biological hallmark capabilities with distinct circuits that operate within cells. And in one sense, this is a fool's errand, if for no other reason. We really understand this intracellular circuitry very incompletely. In fact, so incompletely that we really can't predict precisely how a cell will respond after it receives, a normal cell, certain kinds of afferent signals. You probably know people in bioinformatics and in artificial intelligence who say, just give us this circuit diagram and we'll be able to predict for you exactly how a cell is going to behave. Obviously here, each of these green circles represents one or another signal processing protein. If one of these people tells you that they can predict exactly how a cell behaves on the basis of what little we know about this signal transduction circuitry, you need to regard that person with great suspicion. Or as I often say, if you shake hands with such a person, you should count your fingers afterwards to make sure they're all still there. They are not to be trusted. We are still light years away from being able to understand how this intracellular circuitry operates. To be sure, there are 11 or 12,000 proteins that are concomitantly expressed in an adult human cell, and how they interact with one another in terms of signaling is so complicated that it's almost as complex as how many stars there are in the universe, at least as viewed at present. Still, it would be a desirable goal of cancer research to one day be able to plot out these distinct cell biological attributes of cancer cells in terms of distinct signal transduction pathways that operate within individual human cells. And to extend that, it would also be very useful to know whether this changes at all in pediatric versus adult tumors. To my mind, there actually are going to be some profound changes in the way by which signals are processed in pediatric cells or fetal cells, and that shifts as these cells move from the fetal to the adult state. But at present, neither I nor anyone else can demonstrate that to you experimentally. I'm thinking now that we have about 10 more minutes to go, and maybe I should begin to solicit some questions rather than continuing. Dr. Resnick, what is your advice?
So one, I'm enthralled by your presentation.
Thank you.
Which I suspect others are as well. So I hesitate to stop you from continuing. The way we have it actually set up is we have a chat channel and a Q&A channel. I think most people are currently watching your presentation, and people maybe can begin putting any questions they might have in those chat or Q&A boxes, and then we can initiate Q&A in a couple of minutes. So if you'd like to continue for a few more minutes while people...
I'll do just that, and then we'll finish. Yep. Here is one additional complexity that we all wrestle with, and that is the heterotypic intercommunications between individual cells within a tumor. As you know, tumors are not formed just from cancer cells. There's a whole series of ancillary cells, the so-called stromal cells, and we don't really understand most of what happens as cancer cells interact with their surrounding microenvironment, as is indicated here. This represents yet another dimension of complexity. One is making significant headway here, in contrast to the apparent almost no headway being made, no progress made in trying to understand the signal transducing circuitry that operates within individual cells and somehow is able to tell cells whether or not they should proliferate or not. And by the way, as you may know, young generations of cancer researchers are increasingly less interested in doing signal biochemistry, protein biochemistry, and therefore the prospect of trying to understand the signaling circuits within individual cells is receding further and further into the distance rather than becoming more and more plausible. here is yet another depiction of this complexity, because here, if we deal with the parenchyma, which in this case operates in the cancer cells themselves, and the recruited stromal cells, both innate and adaptive immune cells, as well as connective tissue cells, these interactions change progressively as cells evolve from normalcy to malignancy. And so here we have yet another multidimensional area of complexity, which is also very poorly understood. I finish finally with one sermon, which I think I like to preach, which is the following. I've tried to suggest that somatic mutations are critically important for the biology of cells, as I preached early on in today's talk. The question is, how important are non-genetic factors in determining the biology of cancer cells? And here I give you two instances where non-genetic programs are very important. If we look at the normal cells of origin of a tumor, here's an experiment undertaken at Stanford, where two very closely related cell types in the lungs acquired the same set of engineered mutations. And they both generated tumors, they're indicated here. And these two types of tumors from closely related lineages in the lungs of identical genotype had very different transcriptomes, as indicated here in this volcano plot. That means that, in fact, the differentiation program of a normal cell of origin obviously influences normal epithelia. But in addition, this differentiation program imposes itself on the biology, not only of the normal cell, but on all of its subsequent descendants, which are increasingly perturbed by the acquisition of various somatic mutations. And at present, we know almost nothing about how somatic mutations, which I've indicated here, interact with the pre-existing differentiation program of the normal cell of origin. Here's another non-genetic process, and that is invasion and metastasis. Just to finish up now, the fact is that invasion and metastasis is not choreographed by mutations. In fact, if we look here at this process and move from primary tumors to distant invasive and metastatic derivatives, this is a complex process called the invasion metastasis cascade. And at least in the case of carcinomas, it's all choreographed by a cell biological program, an epigenetic program, which is called the epithelial mesenchymal transition, and has a whole series of cell biological attributes as listed here, which are acquired not because of genetic mutations, but because of changes in the tumor microenvironment, the stroma of an evolving cancer cell. As first described in this case by Betty Hay, Elizabeth Hay of Harvard Medical School, or as they call themselves in Boston, the medical school, much to the distress of the other ones in Boston. Okay, let's have some questions. Maybe I'll get out of my PowerPoint and try to solicit questions. Or maybe Dr. Resnick could read the questions to me if he has access to them.
I had a couple questions that came my way. The first one was, given the framework you described and the fewer alterations genomic present in pediatric cancers, are there predictions on their targetability? So what I think what the person is asking here is, there is this concept that the more mutations you might have, the more challenging it might be to target a particular cancer. And so the question is the inverse, or what's your framing for the
it's an interesting question. I cannot answer it. I really I don't have a good feeling for the extent to which the targetability therapeutically of cancers is relatable to the number of mutant alleles they acquire. It's a very provocative question, but I cannot provide an intelligent or even unintelligent response to it.
Perfect.
Hey, Adam, can I interject with a non-chat question, but I think something that might be a useful perspective, Dr. Weinberg, given that we have a mixed audience, and with our CBTN goal is we are trying to accelerate the pace of, pediatric brain tumor research. So not so much brain tumor specifically, but as a leader in cancer research, and we have young investigators and some students and some people who are still in training on this call, what would be your advice for an early career scientist or a student to an aspect to study or a way of thinking about cancer based on your experience?
Well, I think the hallmarks of cancer is a useful way of thinking about cancer, but I don't think it's necessarily a particularly good instrument for focusing one's own career. One has to focus on an interesting question. To my mind, the most provocative question I raised today was, why are pediatric tumors biologically so different from adult tumors? That's a major unsolved problem. Nobody really has a good answer to that. I believe that the biochemistry of signal transduction shifts as pediatric cells or fetal cells go into the adult mode. And whoever figures that one out will be a candidate for an invitation to Stockholm, to my mind. I think that's a very exciting area to work on. But if you ask me how to start with that, I haven't the vaguest idea. And I'm just being presented by a cosmetically much more acceptable set of glasses. So my wife says I look more intelligent in these glasses. Okay, anyhow, anything can happen in the wilds of New Hampshire.
A second question that came up, and I think before I read it, a large amount of our consortium's efforts has to do with, studying from the perspective of measuring the biology of tumor samples themselves as, biosamples collected across the multiple institutions and creating data sets that measure different elements of that tumor, whether it's mutations or expression, or as you highlighted, single cell analyses. Given your warning around the challenges of putting the pieces together, what would be your framework for addressing the solutions of integrating such data sets with the laboratory-based biochemistry or model system-based analyses?
Well, here, if we're going back to admonitions for young investigators, I would say that to my mind, people have fallen in love with generating enormous data sets over the last decade, wildly beyond our ability to digest them. If you look at how many useful take-home lessons have emerged from most of these data sets, you will be rather amused and disappointed. So to my mind, what's useful for a young beginning investigator is to focus on a discrete and specific biological question, and not simply to gather enormous heaps of data, which neither they nor anyone else will ever be able to interpret, that will certainly be archived on some library shelf somewhere, but will never really accrue to our increasing understanding conceptually of how cancer pathogenesis operates. So I'm very much focused on concepts rather than the quality of science being improved by gathering large amounts of data and pooling data and doing meta-analyses and ultimately coming up with correlations which have little compelling merit in terms of understanding the mechanisms of pathogenesis, which I think are so critically important.
Right. Let me reframe that and see if you agree with my interpretation, because the CBTN is definitely a data-driven organization, but I think your admonition. So you may or may not have heard Angela's introduction, where she lamented the number of hours she spent in cold rooms and laboratory benches doing experimental science. But maybe let me ask you if the following framing might be relevant, which is that data on its own is insufficient to drive biological understanding, and that the intersection between the experimental hypothesis-driven questions that you need to address have to intersect with those data resources, and that independently of that process, you can fall short. Would that be a fair characterization?
I think I agree with that entirely. Lots of young people these days want to go into the area of bioinformatics because they say it can actually distill large data sets into relatively small, useful take-home lessons. But if you look at the reality, the fact is that there still needs to be people like Dr. Wanders who went into cold rooms and who ran gradients and chromatographies to figure out what's actually going on. And although there will be many bioinformaticians that will arise over the next decade, I don't know how much useful information they will provide relative to the few people who survive and, like Dr. Wanders, go into cold rooms and actually do experiments. Those experiments, to my mind, will ultimately be more telling than analyzing vast data sets with the hope, to my mind, often very unrealistic, of being able to reduce the information in those large data sets to useful insights.
So Dr. Weinberg, if I can post a not contradictory comment, but I guess just a way of thinking of it. So as a physician scientist, the reason I went into the bench and I spent time learning molecular biology, injecting mice with tumors, were the clinical observations that I made that I really felt from a biology standpoint needed to be interpreted and sometimes from a clinical lens. So a huge part of our efforts have been not data generation just for the sake of data generation, but to inform on the biology we are seeing clinically. So from that aspect, not just generating data to generate data, but if you have rare diseases in which you need to power your statistics to be able to answer some of the questions and actually go back in and do the additional laboratory work. Because I will say, when I was early on in my career, and unfortunately, I can no longer be considered an early career physician scientist.
To my mind,
what I found really, really challenging was having enough data for validation and or to explore further hypotheses. So a big part of our data generation efforts are actually what happened to the patient.
Well, I understand that. And obviously, we need to know how clinical responses happen or don't happen. But I still argue that preclinical research, and when I include or I mention preclinical research, I'm mentioning not only going into the cold room, as you bravely did, but animal models and cell culture models and a whole series of models which are inspired and provoked by specific mechanistic questions. To my mind, we will never understand why certain patients respond and others don't respond unless we understand at the mechanistic level what it is that governs their responses. And the bioinformaticians will, in very large part, I wouldn't say never, will never be able to throw us a lifeline here. Somehow, preclinical experimentation will need to be definitive in determining and informing clinical choices. And that need hasn't gone away. Some people live with the illusion that no one needs to do any more of these dirty hands-on experiments anymore. We moved beyond that. And I fear that's a terrible illusion.
So I think we're of shared mindsets, because I think, our goal or our concept was to not have data generation be a rate limiting factor, but to allow these resources to be democratically shared, so that, just like what you're saying, people can go back into the labs and answer important questions and not have to spend their time and resources on data generation just for data generation's sake. So ultimately, I think we are saying the same things. And I think your perspective, especially given, I'm sure you have seen things that become popular. Everyone jumps on that bandwagon and are forgetting that they still need to go back to the bench and answer and ask some of the hard questions in a methodical, scientific way in a preclinical modeling.
You said all this in a much more articulate and diplomatic way than did I.
And I also have to thank you, Dr. Weinberg, for your encouragement of laboratory-based primary research. I still have carpal tunnel syndrome in my thumb despite the fact that I have not been pipetting for a very long time. But
You look okay.
that's true. But that to your point, it's a connection between the experimental framing at the laboratory and the interpretation of large-scale data set that needs to occur. Some of our staunch challenges remain, which is we don't have enough laboratory-based researchers focusing on the pediatric question at hand in the way that you described.
Yep.
Our goal is to actually frame those questions tangibly in ways that entices and drives the new generation of researchers to ask those questions and address them clinically.
Adam, can I end with one last question from the chat before we take probably just a 5-minute instead of a 15-minute break? Dr. Weinberg, one of our people asked, given the epigenetic, genetic, and microenvironmental heterogeneity of brain tumors, and especially pediatric brain tumors, are you surprised that we actually do have some success in curing some children?
Well, again, I have no preconceptions as to what leads to cures. Mm-hmm. And for me, I cannot explain why some tumors are curable and others are not. And I don't know whether many of them, some of them like neuroblastomas obviously would have gone away on their own, but many others are directly attributable to your therapeutic intervention. But having said that, I will defer from saying anything about clinical responses. I just don't have very good insights into what governs them or the lack thereof. Now,
but I think you do believe in cancer research and the hope that we can, as a community, not cure all cancers, but that we can find by focusing on the right questions.
Every step at a time.